the address. So now I would like to start our second poster session, where we have 8 posters in addition to the poster specials that you had the pleasure of hearing in more detail. So I would like to start, if possible, if Miss Dr. Cui is here from China.
Hello.
Cui-san. Nice to see you again.
Nice to see you. Can you hear me?
Yes, I hear you perfectly.
Okay.
Dr. Cui is actually also a little bit special among our poster presenters. She is a winner of the poster award at the previous conference. She is from Professor Xiao's laboratory and doing fantastic work about on aquaphotomics using temperature perturbation. So, Cui-san, can you tell us about your poster in few sentences? Can you tell us what you did?
Okay, I'll share the recorded presentation, and then I can introduce my poster. Okay. Can you see the video?
Yes.
Okay.
No sound.
No sound?
Yes.
Okay, I'll check. How to do that? This will be okay. Maybe I can just introduce this for all of you.
Yes.
Okay. Hello, everyone. I'm the postdoc in Peking University, and during my PhD study, I'm the PhD student in Professor Xiao's lab. Today, I'll share the topic is temperature-dependent variable selection in near-infrared spectra for aquaphotomics. We all know temperature-dependent near-infrared spectroscopy has been developed to analyze water structure in aqueous system and taken as a new technique for aquaphotomic studies. It has been used for many studies like water and protein formation, and for protein role, and for illustrate the protein and the role of water structure. However, due to the complexity of water characteristics, spectra in temperature-controlled near-infrared spectroscopy, it's difficult to obtain information from the spectrum to understand the effect of temperature on water structure. In order to investigate the temperature dependency, this study proposed a method to select the temperature-related variables from the temperature-dependent NIR spectra. This study made the experiment first by the simulated data. They use a Gaussian function for the simulation. First, we just simulate a Gauss peak, and then made the simulation of temperature by increase the intensity of the Gauss peak. Then we use the continuous wavelength transform to make the multiscale was stopped to account for the spectral information with different frequency, which can be viewed as a derivative of the spectrum with different smoothing window with this sub-picture. And then we use the macro MCUVE method to select the variable with the temperature dependency. This is a method to build the temperature and the spectrum for the just like PIS. And then we can see this is the result for the MCUVE. And we can see the main peak is in the consistent with the standard deviation of the spectra. Then we use this method. We can continue with this method to show the application with the real spectra. First, we use it in water. We can see water spectrum is more complicated than the simulation spectra. And then we use the method. We can get this picture. It's like a fountain, spread the water spectrum with the features. So we call it the fountain graph. We can see from the fountain graph, there are more spread, and we selected more peaks, just like the water graph patterns. And then we use it for the sodium chloride, glucose, and HSA solutions. The spectrum of this is also variated by the temperature. Then we use this method, we can get the graph fountain graph in the sub-feature. We can see there are little difference between the water spectrum and the solution spectrum. In order to make a more clear comparison, we just cut the scale for 30 to see the difference. Then we got this picture. We can see the selected peak, also call it the selected variables. In the figure, we can see water and other solutions. It's a little different, but in the same range. This difference may be this difference may be caused by the solute on the water structure. And then to verify the method, we also use it for a quantitative models. We use it with glucose solution spectrum. We choose the peaks, like the 7 peaks, and the intensity of these peaks to do with the temperature and temperature and the concentration. We see the intensity of, let's see this peak with the concentration. It has a good model for quantification. In summary, this method for selecting a temperature-dependent variable for the temperature-dependent NIR spectra was developed, combining CWT and MCUVE. The selected variables are the reflection of the water structure in aqueous solution, and thus the variables are informative to discriminate the samples. The method may provide a tool to identify characteristic variables in NIR spectra for understanding the function of water in aqueous system. Thank you.
Thank you, Dr. Cui. So anyone has a question regarding the poster of Dr. Cui? I just want to say we have time for discussion until 3 p.m. So any question, you can also write it in the chat box. Privately, of course, to Dr. Cui.
I have a question, Jelena. Is it okay? So thank you very much for, very nice to see you. again.
Very nice
Dr.
to see
Chen.
you.
Congratulations.
Happy retirement.
Thank you. Congratulations on your doctor degree. And I hope you enjoy your new work and continue working with Professor Xiao's group, right?
Yes.
I like very much when I read your paper, actually, I found the idea about the fountain graph really, really wonderful. And I have one kind of question. I see that you, and most of people do like this. So when you do different temperatures, people usually choose 20, 25, 30, 35, 40. But if you go to papers, you will see that water has nonlinearities exactly at those temperatures, like 25, 30, 35, 40, peculiar nonlinearities. And I would suggest to, because you work a lot with temperature, and this is for Professor Xiao as well, I think he's listening, to see what exactly is happening at those temperatures. Because we have tried in my lab, and I particularly try to use in between, like 34, 36, 38, kind of, to avoid those nonlinear points. But on the other side, it's very interesting if you stay there and see what's happening. That's my suggestion. And number two, I've been thinking, reading it, because I'm really, it's immense work what the whole group is doing. It's very, very important for science and infrared and aquaphotomics. In aquaphotomics, what we try to do is to bring people working using water as a mirror in order to understand what is the role of water for functionality in systems. So we need a unique language. We need the same language for everybody in order to understand each other. There are so many disciplines like physics and chemistry and spectroscopy involved in this data analysis. But if we try to establish one language, common terms, then people will start learning those terms, and then we can communicate between us. We can understand each other. So I would suggest in your papers, presentations, if you use WOMAX, water spectral patterns, the terms that we already have been using in aquaphotomics papers, then it will be easy for people to use your technique, to understand, to multiply, and to speak the same language.
Yes,
Thank
yes.
you.
Thank you.
Thank you.
Thank you, Professor Tenko. I have a lot of questions, but I'm talking too much. So I will, I hope I can talk to you a bit later.
Okay.
If from the audience nobody has a question, well, in that case, Dr. Cui, thank you very much. I think this is very, very fantastic work, and I hope you will continue to other regions of the spectra using the same method to bring them, and especially around the temperature 37 and around the temperature 4 degrees. I would love to see that you work with the substance that you work at this temperature. Okay, thank you once more. Now I would like to ask, is here Sukrita Ananta Vitayanon, called Brim for sure, for so short. Brim, can you please share your...
Can you see me? Yes.